Hello everyone, I am Rajeshwari Moganty, and today the module we are talking about is on the introductory molecular biophysics, and this paper is on the biophysical techniques which are devoted to spectroscopic methods. I am working at the All India Institute of Medical Sciences at New Delhi, and I would be giving you a brief overview of what we are going to study in this particular paper on the biophysical techniques. So the objectives of this module is actually meant to give you an overview of different spectroscopic techniques and also what are the basic principles based on which these techniques are actually based. So we will be discussing each one in a very brief manner. Also, the third main objective is to understand the application of these in structural biology and molecular biophysics. So, with that objectives, laying out those objectives, I would like to give you an introduction that actually the biophysics techniques can be studied in a number of ways, and these techniques are based on different chemical and physical properties of the biomolecules. So, each technique actually exploits one single property in order to understand the conformation of biomolecule, like for example protein and nucleic acid. Now, the first section is actually devoted to different microscopic techniques, like for example which are usually used by the biologist: light microscopy, electron microscopy, transmission microscopy, scanning microscopy. Now, the second section we will start with the various absorption spectroscopy, UV visible, then circular dichroic spectroscopy, fluorescence, and so on and so forth. So, whereas these techniques are actually mainly, the main purpose is to understand the structural organization both at cellular level and molecular level of all these biological systems. Now, the molecular spectroscopy is actually, what is it? It's a study of interaction of the electromagnetic radiation with the matter. If based on these things, we have made 32 modules in this paper. Now, of course, one is introduction, what we are going to give you now, and then comes the light microscopy, transmission and electron microscopy, then comes the scanning microscopy. So, after the microscopy techniques, we will study what is the electromagnetic radiation and how does it interact with the matter, for example, various biological molecules or the biological systems, proteins, nucleic acids, cells, and so on and so forth. The sixth chapter is on the UV visible absorption spectroscopy. There you will study the famous Beer-Lambert Law, how the instrumentation is, what are the principles and what are the basic fundamentals of these absorption spectroscopy, particularly to study the proteins, amino acids, and so on and so forth. And also commonly used, how do you determine the concentration of a protein using absorption spectroscopy? It is the most common used in every day in a biological laboratory. The next chapter is on the replication, DNA replication. Before studying, you have to understand how the DNA structure, and then comes the application to understand the DNA, RNA, then estimation of the concentration using 260 absorption, and also use the purity of the DNA and RNA. The next chapter is of course on the Helix-coil transitions, how do you determine the TM, the melting temperature. Then after studying both molecules individually, you will study the DNA drug interactions. Then the fluorescence spectroscopy is dealt in a very detailed manner, where all these chemical structure, what are the factors that actually the fluorescence depends on, the medium, temperature, etc., what are the quenchers, and so on and so forth. Now, later on, the fluorescence energy transfer is only in an exclusively studied to understand the polarization and so forth. Then later you apply these techniques, methods to the proteins. Then how is a ligand polymer complex is formed, and how do you understand that, then the scatter plot, and so on and so forth. Then again, after studying different, understanding the binding of two molecules, whether it is a weak interaction or a strong interaction, or weak and strong, how do you differentiate these things will also be studied. Okay. Now comes the luminescence. The bioluminescence is one important aspect. When you understand, when you study fluorescence, you cannot ignore bioluminescence, chemiluminescence. Then how do you label the biomolecules which are actually not fluorescent? Okay. Then comes the green fluorescent protein, one of the most popular one in biology that is used. Then determination of the protein structure, secondary structure, then by using the infrared spectroscopy. Also the tertiary structures you determine near UV, protein denaturation and so on. The next module, the conformation of nucleic acids, drug DNA interactions, thermal stability of nucleic acids. Then the later part is on the IR and Raman. The 24 module is devoted to IR spectroscopy and vibrational frequency. What are the types of vibrations you come across in this? What are the dipole moment and so on and so forth. And then also when you finish the IR spectra, you have to understand the IR spectra, what are the different functional groups which are also called thumb rules, which are used to study the proteins and peptide groups and so on. So there is after that advanced technique, that is the FTIR, which is given on, which is devoted on module 26. Then you have Raman's, Stokes, Raman spectra, resonance Raman spectroscopy, also the Raman spectra of the proteins, which is important. Raman gives a different kind of information as far as protein structure is concerned. Then of course atomic absorption spectroscopy, flame photometry, which are usually used to understand the elemental analysis. Then you have the light scattering experiments to understand the structural aspects of the whole cell. Then SPR, or the surface plasma resonance spectroscopy. So after studying the light microscopy or the optical microscopy, we will do the transmission microscopy, electron microscopy. Actually here it is a very important technique to understand the structural organizations of the cellular organelles. And this specimen preparation, you will see how to make the specimen for the TEM, that is popularly known as TEM, and the most important application is in the infectious biology, also cancer research, material sciences, nanotechnology. They are all very important techniques to understand. So the TEM and how are the SEM different, that is a scanning electron microscopy. So the next is the scanning electron microscope. Now this is again with a small introduction to the scanning electron microscopy. What is the working principles based on which this is developed, and this is actually used mainly to elucidate the structural organization in a cell, and also the main purpose is to use it as an application to understand the surface details of the microorganisms. For example, the cellular, even if it is a cell or a bacteria or any tissue, to overall cellular organization. And the main difference between the TEM and SEM is also important, because each one has its own advantages. Now the next one is on the how the electromagnetic radiation interacts with the biological systems. Here I want to introduce you to the basic concepts of the parameters that actually are related to the EMR. You did study these things in your paper 1 and also in paper 2, which is completely devoted to quantum biophysics. However, it is important when you are studying the interaction of these electromagnetic radiation with biological molecules. So this has a different kind of a approach to understand the molecular structure level. So some amount of basics will be a kind of repetition, and nevertheless it is meant to understand the polymer structure, that is a protein and nucleic acids. Now then also will interact with these biological systems and understand enumerate various biophysical techniques based on the absorption, scattering and so on and so forth. Now the UV visible absorption spectroscopy is the most fundamental one, where you will be studying the famous Beer-Lambert law, and then in description of these instrumentation, both the mono this thing with one beam and bi-beam with two beams and so on, various techniques involved, technical details involved in this instrumentation, and then of course the application. Now there is understand the UV visible spectrophotometer to practical problems also. Then you study with the peptide, what is the contribution of peptide in a protein, then aromatic amino acids, how, which range they absorb, and what is application to determine the concentration, and so on and so forth. Prosthetic groups, those proteins which have an additional absorption other than the peptide bond, you have prosthetic groups like heme group is there. So that will also contribute to the absorption spectrum. These will be discussed in a very detailed manner, and role of the disulfide linkage and so on. Also the prosthetic groups in proteins, we have more differentiation, more in a detailed manner, and determination of the pK value of the amino acids like tyrosine, and where are the aromatic amino acids, what are the conformational changes in proteins, protein folding, protein ligand interaction. Also very important is enzyme kinetics we will be studying. Then the other chapter is on the module is on the protein conformational changes. Nevertheless, here you will be using in a much more detailed method that is the for concentration determines Beer-Lambert Law. But again you will use the understand the conformational aspects. What are the other factors that determine the conformation of a protein, whether it is a folded or a denatured structure. All these things can be very easily can be studied using a simple technique, that is UV absorption. Then one chapter which we have devoted to the replication of the DNA mechanism before we study the various models of the DNA, understanding the like various Messel shaft experiments, prokaryotic DNA replication, what are the basic mechanisms involved, proteins involved, eukaryotic DNA replication. So this gives you an overall picture of how the DNA replication occurs. And then of course you will study the application of the absorption to understand the DNA structure and RNA structure, and also use that how the bases contribute to give the absorption, and how do you determine the concentration, DNA purity, how much is the protein to absorption. 260 by 280 is a very common parameter that you determine, and discuss about the absorption but the various nucleic acids. There are also backbone contribution, how does it influence the absorption, and then of course determine the concentration to estimation of the concentration, absorption spectroscopy for purity of the nucleic acids, and so on and so forth. So after studying the proteins and nucleic acids, absorption, also determination of these homogeneity of the protein and DNA melting, helix to coil transition, like in protein you call folded to denatured, here you call it as a helix to tail concentration, effect of temperature, concentration, and so on and so forth. So now you have studied the fluorescence basic principles and application to molecular biology, I mean molecular structure. Then also you have studied the the bioluminescence. But then comes the significance of these fluorescence. If there are, we have to label these molecules which have for understanding the studying the distance between them, and then you use the enzyme labeling and also tag them with the different kind of fluorescent labels. And sometimes we use the commonly used fluorophores and their mechanism, how do they bind, analysis of the biological processes through protein labeling. All this will be studied in the labeling part of it. Now the next thing is a green fluorescent protein, which is a very the biologists are very much fond of this. This is very important because the discovery of this milestone happened in the because of the GFP protein, and GFP chromophore gives a different kind of an environment, and understanding the mechanism of the fluorescence actions, the GFP as a marker for the gene expression is the most commonly used application. GFP in protein targeting approaches, and also GFP as an active indicator of cellular activity. These will be discussed in the module 20. Now after doing that, I'll shift the gears now to a new technique, that is the circular dichroic spectroscopy. This is actually an absorption spectroscopy, but here the light is used is the circularly polarized light, not the unpolarized light. Now in this you will also see what is the difference between optical rotation, dispersion, and the CD spectroscopy. Then you will study the information available from the CD, how to understand the proteins, which gives an additional information compared to that of the what you gain from the UV or absorption spectroscopy. Then the next chapter is to understand the protein structure using a different kind of the electromagnetic radiation, that is the infrared radiation. So here you have near UV and far UV. Now how UCD is used to understand the structure, secondary structure of the proteins, alpha helix, beta sheet and so in the UVCD region, and also the tertiary structure which you study in the far UV region of the CD spectroscopy. Evaluation of the conformational changes you will get also from the CD spectroscopy, and you will use it also for the equilibrium and kinetic experiments. As far as the nucleic acids are concerned, you also have these CD spectroscopy which gives a different kind of information. The next chapter is on the nucleic acids, because they are not naked, they are all the time interacting with drug molecules, small molecules, ions and proteins. So this is one of my favorites also, the interaction of the nucleic acids or the proteins with other molecules. So what are the factors that actually depend on the DNA double helix structure, which is accepted as a which exists in the B conformation, but actually it depends on a number of parameters, like which are the small molecules present in the environment. So it is important whether it is a drug or any other small molecule or a metal ion. So these interactions you will be studying in this chapter. Now the IR spectroscopy or the vibrational spectroscopy. What are the types of vibration spectroscopy, and whether there is a homonuclear atoms are there in the molecule, or a molecule is a heteroatomic structure, how does a vibration different, and so we will be giving with the small examples, and then of course we will move on to extend the same thing to understand the IR spectroscopy to biomolecular structures. In this you will be given more of details about the how an IR spectrum is designed, what are the significant peaks in it, assignment of these peaks and all you will be studying. Then IR of the particularly of the peptides and proteins, how does an IR spectrum, which is a thumb rule, you also see the different peaks which come from different regions. So how do they look like in a protein? Then comes the IR spectra of the biological macromolecule. After studying the proteins, you go into the polymers, biopolymers. Then comes the FTIR or the Fourier transform IR spectroscopy, because a simple IR is not useful for the understanding the biopolymer structures. Only if it is a small peptide it's fine. So FTIR is a very boon to understand the protein structure in a different manner. Then of course you will study the scattering, elastic, inelastic scattering, Stokes methods, Stokes scattering and so on and so forth. Then comes the resonance Raman spectroscopy. Then the Raman spectra of the protein. Raman is also actually comes under the IR region, but Raman is different from the conventional IRs or FTIR. So what are the Raman spectroscopy, what different types of Raman spectroscopy, and how is it useful, what are the applications, what is the advantages of this over the IR, and what is the limitations of Raman over IR. So these things will be studied in a very comparison with the IR, and what are the factors that influence the Raman spectra of a protein. So this will be discussed in a thorough manner, because Raman is one of the very important spectroscopic techniques to understand the molecular structures. So after doing that, we will jump to another region, that is the atomic absorption spectroscopy and the flame photometry. These are actually for elemental analysis rather than for a polymer structure. So here we will be doing what is atomization, how do you get these, how do you make them using the flame photometry, and what is its application mainly, even though it is not so popular as compared for structural biology point of view, but nevertheless they are important. Then light scattering, which is very important. Then you have SPR or the surface plasma resonance spectroscopy. So these are all the techniques we have tried to put them in an ensemble in this chapter or the paper on spectroscopy based biophysical techniques. So I think to give a overall view of these modules studied in this you will be introduced, will be doing it in this paper, is something very important in a biophysics point of view, structural biology point of view. So also you will be doing to how to detect the interaction of these carbohydrate protein interactions, because carbohydrate per se does not have any secondary structure like a protein or a nucleic acid, but then carbohydrate, when it binds to a glycoprotein or binds to a DNA, or how does it actually interface with these things, will be studied also. We will be dealing with these things also. So at the end, what I would like to summarize this is that what I would try to explain is that what are the topics that are being covered and what are their justification, that why you need, as a biophysics person, as a structural biology person, why do you need to study these techniques, is something everybody will has to ask before studying. And therefore in this module I try to make you appreciate the need for each technique, the need for understanding the molecular structure of a protein or a nucleic acid from different approaches. Each technique gives a different kind of information, although it is the same protein molecule. So IR gives different Raman spectra, then comes CD, fluorescence. So they are commonly used, but then of course each one gives different information, so that you can get a overall structure of the protein in a very larger perspective. So with that, I hope you would enjoy this paper as much as I enjoyed making it. And you will be dealing with each, when you open each module, you will study in much more detail. Thank you.